So the Early Career Award, as you might know, there have been quite a few over the past years. So it started in 1981. And since then, we have had a long list of candidates. It was called the Young Scientist Award up until 2019, after which we changed the criteria a bit to accommodate more candidates. So this year's early career award, is going to go to Manosji. It's actually a pleasure to introduce it to someone from Belgium. As you know, I'm also Belgian. I had nothing to do with this. I only passed on the nominations to the award committee and I have no say in it, but it's still my pleasure to hand this to Manosji. He was nominated by Professor Lode Goderis, which you all might know. He was on the award committee, but I have to say that he wasn't voting in for him, so he had a conflict of interest there. But he did say in his nomination letter that there are plenty of arguments to award Manosji both on a scientific as well as a personal level. I'll quickly share a few facts and achievements of Manosji with you. I'm not going to dwell too long, not to run over, but just to let you know that there's certain criteria that we always look at. Of course, there's an age limit, the date when you got the PhD can't be more than 7 years, and so on. And then also your PhDs here, sorry, and also the interactions with the EEMGS are also looked into, and I'm pleased to say that he also recently joined the Belgian EMS committee. Yeah, this is maybe a lot to read out now, but just to let you know that the committee really voted for him as one of the better candidates. So these are a few of their appraisals. Both giving him credits for his scientific maturity and impact, as well as other achievements that he did, both like in recent European projects where he is acting as work package leaders. His work mainly focuses on nanotoxicology, but both ecotox as well as genetic toxicology, and linked with epigenetic modifications. I'm not going to dwell more about this because I'm sure he will tell you all about that himself in his lecture. He had also an impressive list of scientific publications, as you can see over the years, and was involved in 3 book chapters. Other than that, like I said, he was involved in various national and international networking projects. He received 3 postdoc fellowships, was involved in Cost Action DemoPack, and is currently work group leader and co-lead in 2 Horizon 2020 projects, which you can see here. Apart from several travel awards, he also received 2 Young Scientist Awards, both in Thailand, and one from ICAM when there was the meeting in South Korea. And he also acts as reviewer for short-term grants for ERS, so the European Respiratory Society, and he's also an editor with the journal Nucleus. And he co-supervised 4 PhD students so far. But other than that, these are mainly scientific achievements. Like I said, Lode told us that there's also quite a lot of personal qualities that make him nominate him as an Early Career Award winner. He says he's a good collaborator and a team player, but there's also some other things we want to share, and his colleagues wanted to share. There's one trick with Manosji, apparently, and the key is coffee. So sometimes he needs the coffee to turn into a more pleasurable and fun person. So everyone knows him like this. The fun person that shares his ideas, is always very helpful and friendly for his colleagues. But apparently the key to that is a coffee in the morning. Otherwise, he apparently turns grumpy. I never experienced it, but I've been told so. Other than that, I'm told and I've experienced that he's a very friendly person. He is really caring about his colleagues, people, but also the family of his team and so on. So you can see here some snapshots of the group going out. He's always there to enjoy time with him. And as you can see, he's always the one taking the pictures. Actually, he loves taking pictures. He also has a hobby of taking pictures in the herb garden, as well as some architectures, buildings in Leuven, where he is living for his work. And it was hard to find pictures from you on your own and not as a selfie, Manosji. So, but we did find one. So with this, I want to say that the floor is yours. Again, congratulations with this award. As you see, everyone supporting you, the team, and we look forward to hear about your work over the past years. So thanks again, and I'll hand over to you.
Thank you, Sabine. As all of you, getting a bit emotional, but as all of you can see, it's with such wonderful colleagues, it's never that hard. Of course, researchers always like writing grants and fighting, struggling for it, but with such wonderful colleagues, life becomes much easier. I'm grateful for that. I will start sharing the screen. I hope you see my screen now.
Yes, we do.
So thank you. The title of what I would like to present is toxicity of carbon nanotube and what I have learned, but more importantly, what we have learned as a collective. And as you can see, the sentence is incomplete and there are lots of dots, which represents the state of how the field is. But before I start, I would do it a bit different, as in I would like to acknowledge a few people, some of whom are here, without whom my PhD and my postdoc wouldn't have been as wonderful a journey as it has been. I owe a lot, and rather a majority of them, to my PhD supervisor and my professor from university, Professor Anita Mukherjee and Dr. Momita Bandopadhyay, both of whom are in the audience, and I'm very grateful for them for giving me that opportunity and seeing that spark in me to continue doing research. Professor Lode Guterres, who was just by sending a mail welcomed me into Belgium in his team, and Professor Peter Hout. And there are several colleagues that I've worked with, both PhD students, researchers, postdoctoral researchers, and I must say thank you to all of you. As you can see, the timeline, I started my master's in 2006 and started my PhD in 2009. Finished it in 2014, and that's more or less the timeline of the topic that I would be talking about, nanotoxicology. 2008, there was a rather milestone paper on carbon nanotube and how it's similar or possibly similar to asbestos from Poland. And that's where I, together with Professor Anita Mukherjee, started working on nanoparticles. We went to a conference where there was discussion on how nanoparticles can be regulated and what are the options and what's known and what's not known. A lot at that point were not known in terms of toxicology. And I'm afraid to say that's more or less the case even today. There has been some improvement in our understanding, but it has taken way too long to first classify MWCNT 2014 as Mitsui 7 as group 2B carcinogen. And I would take you through my presentation, keeping in line this timeline and as we started to gain more knowledge on the topic. Many of us do know what nanoparticles are, so I wouldn't spend too much time on that. They are basically broadly classified as particles which have dimension less than 100 nanometers, defined as either in unbound state or as a group of aggregates or agglomerates which have dimensions in the size range of 1 to 100 nanometers with at least 50% or more particles in that state. And aggregates and agglomerates, I highlight this because that's an important property that determines the toxicity. I will not go too much into it, but there has been some wonderful research trying to untangle this, what's based on primary particle size toxicity and what's based on the aggregation state. As of now, we stand at a market value of around 200 billion euros, which you can, by the number, estimate that it's larger than some of the larger economies even. And more and more particles are being produced. And so it's introduced into the market. Carbon black and silica represents the largest group of those. But then there are other particles of interest for both commercial purposes in pharmaceuticals, including titanium dioxide, carbon nanotubes, and zinc oxide and silver nanoparticles. Now, as we understand the market and the penetration of these particles into our daily life, there has been also wonderful research and summarizing the routes of exposure, primarily through inhalation, where inhaled nanoparticles reach the alveolar space and are distributed through systemic circulation to different parts of the body. Now, from our understanding from where we started, I would like to talk about carbon nanotubes, which are one of the larger groups of nanoparticle, a very complex one to work with, I have to admit, posing a lot of technical challenges with all the assays that are known so far. This is a very generic diagram of what single-walled carbon nanotube would look like. A single hollow tube can be very long, and then multi-walled carbon nanotubes, which are concentric tubes, if you may. Where do we find them? What are the potential sources of occupational or otherwise exposure? There are occupational exposure where they are being produced, but also being used, for instance, in production of tennis rackets or carbon fiber bikes. And then there are environmental sources of exposure. One example would be the case of World Trade Center fire, where the workers who were the firefighters had deposition of carbon nanotube in the lung. But then there are anthropogenic sources where it's found in children in Paris, in school children, and this is what they look like. This is a scary image, and this has led to a lot of questions about how safe these particles are. And because there are no yardsticks yet to determine how toxic they are, we would follow the scale of key characteristics which Martin Smith provided in classification of carcinogens based on mechanistic evidences. There are 10 key characteristics that we can look into, and I will take you through these as we progress through the presentation. And I will come back to this slide to see what we know for carbon nanotube and what we don't. Basically, they can be genotoxic, they can repair the DNA, they can influence the DNA repair mechanism, they can induce epigenetic alteration, oxidative stress, and inflammation, amongst others. Now, the other concern that was continually being raised was about carbon nanotube and its analogy with asbestos because of the fiber shape, their bipersistence, and the lack of breakdown thereof. And added to that, there were concerns related to the nanodimension, which increases the surface area and the surface reactivity, and a lot of unknown about the cellular behavior. Of course, you can see from this image why there were concerns because of the similarity that we can see visually, but also the next picture, which was from 2003, shows from Andrew Maynard from NIOSH of carbon deposition in the protective clothing. And we had to realize that our protective equipments or clothings or masks were not prepared for nanoparticles or fibers even. So what we started off was a study design that was translational in nature. So we had an in vitro study design, animal study, and then study in a human population, very limited in size, however, very significant, I have to say, given the less number of subjects that are available as of now. And this was a study that was the basis of my postdoc research, but also I have to acknowledge this was the PhD research topic of my colleague Denis Donner, and we've worked very closely on all the research that I will show you now. We included classical endpoints of cytogenotoxicity, epigenetics, transcriptomics, and proteomics, and I will take you through the results and show you some of our findings through the next few slides. Now, as we know, in case of nanoparticle toxicology, characterization of particles are very crucial. And we started off with the reference materials from JRC, NM400, and single-walled carbon nanotube, the NIST material, SRM2483. These are well characterized, and we characterize them in our lab as well. But also in terms of the amount of evidence that's there from different European projects, including the nanogenotox project, we thought this would be an ideal choice to study the molecular mechanism. Of course, these are just representative materials that we took and does not cover the entire range of carbon nanotubes that are in market. But nevertheless, we were able to find something interesting out of these two materials. The first observation that we did was basically a visual observation under the bright field microscope, where we, as you can see here on the first figure, asbestos 5 was being deposited, amosite, which we always used as control parallel to the carbon nanotube experiment, and multi-walled carbon nanotube also accumulated here in and around the nucleus. Now, from a bright field microscope, it's difficult to say if it's within the cell. So what we wanted to do was perform electron microscopy, and we saw it going inside the cell, but around the nucleus, as well as in the cytoplasm, as you can see here, shown by the red arrows. But we faced a challenge, as with carbon nanotubes is always the case for all the assays that I will talk about. The diamond knife, every time it passed through the section, it dragged along the carbon nanotubes to find the actual location without intervention was very difficult. However, we can certainly say they are within the cells. So what we used was, in collaboration with University of Hasselt, Hannelore, she helped us with this analysis. She developed a method where carbon blacks and carbon nanoparticles can be detected without specific staining in and around the nucleus. So the green body that you see here is the nucleus, and the red spots are the carbon nanotubes. As you can see here, on the left side is multi-walled carbon nanotube, and on the right side is single-walled carbon nanotube, which are even in some cases piercing through the nucleus. And as you can see here on the figure here, these are the number of spots that we could detect around the nucleus for single-walled carbon nanotube, and it was the highest for single-walled, even at the same dose exposure concentrations. As we move on, we studied cytotoxicity and used sub-cytotoxic concentrations for our genotoxicity study. We used COMET assay to determine genotoxicity. We found significant changes in the increase in tail DNA for single-walled carbon nanotube, which correlates with our deposition of particles. And at the same time, we wanted to see if there is oxidative modification of DNA. So we used FPG modified COMET assay, and we saw significant increase in all the concentrations where there were an increase in the tail DNA over the just the buffer controls for these concentrations. As I mentioned, carbon nanotube interferes with a lot of assays, also the fluorescence intensity, and therefore we wanted to confirm our finding using micronucleus assay. This is the result of a micronucleus assay which we performed in 16 HBE cells, where we saw increase in the percentage of or micronucleus frequency. However, they were not significant. Again, we had to perform both micronucleus with and without cytokalism to avoid artifacts that might arise. And I haven't shown those results in details here, but just to say that we saw a higher amount of micronuclear formation and DNA damage using COMET assay in single-walled carbon nanotube. We then wanted to study epigenetics as one of the mechanisms of epigenetic toxicity. So we used asbestos fibers, chrysotile, amosite, and croxidolites as controls along with this. And at a global level, there were not so much of an impressive change, except for in some concentrations where we saw hypomethylation, so a decrease in global methylation and hypermethylation as well. From there on, we moved on to the genome-wide, epigenome-wide study and transcriptomic study. So we used RNA-seq, and in this case, Illumina 450K array to study the differences in methylation changes. And I'm sorry, the figure is really small, but as you can see here in the axis here, that carbon nanotube group, and also from the hierarchical clustering, that the carbon nanotubes do cluster away from the controls, which was quite reassuring, that given that, and that's the same thing that we saw for the transcriptomic analysis as well. As we moved on to analyze the different pathways that were enriched, we found that p53 signaling metabolic pathways were altered for multi-walled carbon nanotube, but the effects were much more pronounced when it came to single-walled carbon nanotube, which included metabolic activation, endocytosis, and cell cycle progression. We wanted to confirm cell cycle progression using flow cytometry. However, carbon nanotube, as always, blocks the channel for flow cytometry. So a fair warning, working with nanoparticle can be difficult. And the pathways that we found enriched, and as you can see here in red fonts here, they were all affecting intracellular signaling pathways, inflammatory cytokines, transcriptional factors, apoptosis, cell cycle arrest, DNA repair and damage pathway. If you recall the key characteristics that we discussed earlier, these are some of the important ones that were featured there as well. We did similar study, and I'm not going into individual details, but with THP1 monocytes, and there also we saw different clustering based on the uptake, and again, cell cycle progression, mTOR signaling pathways were altered. Similarly, we did epigenome-wide study for asbestos fibers as well in the same cell line, just to compare and have a point of reference. And again, we saw changes in cyclin and mTOR pathways that are being altered for asbestos. Now, we wanted to see the effect of these. These are more effect of, but then a more closer marker to the phenotypic changes would be the proteomic markers. So we wanted to see how they are being altered. And again, we were very happy to see that multi-walled carbon nanotube and single-walled carbon nanotube clustered separately from our controls, and that multi-walled carbon nanotubes were distinctly clustered from single-walled carbon nanotubes. They, as you can see from the Venn diagram here, 172 proteins were significantly altered for multi-walled, while 89 distinct proteins were altered for single-walled carbon nanotube. Looking into, they formed a very complex network of different proteins, including some hub proteins that included p53 signaling as well. And what we did was looked at the differential gene ontology, and we saw significant changes in NF-kappa signaling pathways, synaptic vesicle exocytosis, among others, for multi-walled carbon nanotube. And again, cornification, keratinization, which were associated with apoptosis and cell cycle progression, and a programmed cell death here, as you can see, the ones indicated in red, which showed significant changes for single-walled carbon nanotube. Now, moving on from our in vitro study, and this by no means is complete. So every time we find something interesting, we go back to our drawing board, design an in vitro study, and continue our research on that. But then we also started our in vivo study. I unfortunately do not have the entire result, and I do not want to jump to conclusion before I finish the study. But just to show you something from 1 year after the injection of asbestos and carbon nanotube, these particles are not cleared off. And this again goes back to the biopersistence of these particles. And through histology, you can appreciate how the carbon nanotubes are being deposited. And these are asbestos fibers which still persist, along with the macrophages which are surrounding it. And we are now looking in performing immunohistochemistry as well as other omic analysis to understand what's going on for in vivo. But we were fortunate enough to have a human population, access to a human population in collaboration with TNO, Kerry Leuven, and IRAS, and we were able to characterize exposure for them, shift-based personal sampling, and through general lifestyle and health questionnaires. Campaigning was done. As you can see here, we determined elemental carbon from personal samplers as well as electron microscopy to determine how they were distributed. These are published by my colleagues and are available for you to read. These are very interesting results, I have to say. And we saw larger aggregates of carbon nanotubes that were available, like that were found in the sample site where the workers were there. So they were not individually in the nano size, but they were bundles of 500 nanometers to 100 micron fiber composed of carbon nanotubes. These are multi-volt carbon nanotubes. Based on the elemental carbon concentrations, we determined the mean inhalable concentration. So the exposure was between 4.5 and 42.5 micrograms per cubic meter. And the mean respirable concentration was between 0.07 and 4.45 micrograms per cubic meter. This is far higher than the NIOSH REL, but again, and this is something that we have been talking about and discussing for several years now, that the NIOSH REL is based on measurement limitation and not so much on the health effects. So we still have to establish a safe value based on the health effects that we have observed so far. So this is the study population that was included for the biological sampling. So we had access to 43 matched controls and exposed population of 24 subjects with a cumulative or a mean exposure of 4.25 years working with carbon nanotubes. So that was one of the wonderful things, that these people were working with one type of particle for the last 4.5 years. And that was unique in a way, because in a large number of factories, for instance, in studies that have been published by groups of Lee et al. In South Korea, there are workers who have worked with different metal oxide particles, and to find the exact relationship is often difficult. However, we have to remember that this is a cross-sectional study, so it just captures one time point and not really so much. The ideal case would be to have a longitudinal study. In this, just to summarize, they had a cumulative exposure, as I mentioned in the last slide, and we were able to divide them further into subgroups of operators who had the highest level of exposure of up to 45 micrograms per cubic meter, and there were 7 operators, 6 working in the lab who had higher level of exposure to carbon nanotube at 7 micrograms per cubic meter, and the lab low who had 1 microgram per cubic meter. The first thing that we did, we were not able to perform COMET assay or micronucleus because of sampling issues there. However, we were able to perform DNA methylation studies in DNA extracted from whole blood, and we did not see global methylation changes. And as you might recall, this was the case as was in vitro study. At global level, we did not find significant changes. Then we moved on to study the line 1 methylation using pyrosequencing, and we did not find significant changes here. Line 1, a transposable element, a repeat element, which is almost 15% of our genome, can be a good representation of methylation status as well. However, we did not see significant changes here. But if we then decided to go for sequence-specific methylations of some of the key genes that we identified from our in vitro study. That included DNA methyltransferase 1, histone deacetylase 4, SKI, an oncogene, and ATM, which is involved in DNA damage repair pathways. And for all of these, at least in 1 or more CPGs, as you can see here for DNMT, in the sequence region, we found significant changes in 3 of the CPGs. That's the same for histone deacetylase, and 1 CPG for ATM, and 2 for SKI oncogenes. Now, this was a grouping based on control and the exposed group. So that's 24 exposed versus our control. And then when we looked into the different exposure groups, as you might recall, and here in summarized, we still saw a kind of a dose response relationship for some of the genes, at least. This was quite encouraging to see that there is an association between the exposure levels in these groups. There have not been multiple studies, but on multi-volt carbon nanotube, but there has been a study from the group of Anna Shvedova and from Kazan, and they also observed similar sizes of particles as compared to ours. From our study, as you can see, the exposure levels are within that range, and the respirable fraction are also around the same range here. They observed significant changes in inflammatory cytokines, as has been observed in our study as well, but I have not presented the result because I was not involved at that stage for the study, but also in biomarkers of interstitial lung diseases, which was also seen by the group of ELCO and from TNO. Further on, the group of Fatkodinova and Shvedova, they studied mRNA and miRNA profiles in these workers. We have to keep in mind that these are really a small group of workers, but what we saw was significant changes in cancer-related pathways, respiratory disease, which is something that has popped up in our previous studies as well. And that's in line with what we have seen in our population. We further went on to see, because there is a change, like there is inflammation and there is oxidative stress that we have seen in vitro. We wanted to see if there were changes in mitochondrial copy number, which could reflect the status there. And we did see a significant increase in mitochondrial copy number, and also the same between the groups of exposure. As you can see, operators had the highest mitochondrial copy number. This could be potentially linked to inflammation, but also higher oxidative stress at that point. However, we do not have parallels from nanoparticles. We do have, however, parallels from particulate matters, as well as other compounds such as arsenic and in some forms of cancer. We then went on to study telomere length in the same samples, and we were expecting, as always, we were pre-programmed to think, okay, there would be a shortening of telomere length, and that's what we were expecting. Well, we were surprised that we saw a significant increase in the telomere length in workers exposed to carbon nanotube. And I'm not saying that this is something beneficial. That's not at all the message that I want to provide here, but it's something that has been, and on the contrary, the increase in telomere length has been linked to aggressive forms of cancer, such as leukemia as well. But again, I would not even be alarmist in this case, because it's only a cross-sectional study. A longitudinal study would provide much more information. And as you can see, between the groups as well of exposure, there were significant changes here. So just to summarize here, what we saw so far from the human samples were, they were far exceeding the NIOSH recommendation. There were, of course, observed health effects in these populations, including inflammatory response, changes in non-coding RNA, DNA methylation changes of key genes, and increased mtDNA copy number and relative telomere length. If we go back to literature and see what is known, and this is a wonderful summary from Dong and Ma from Nanotoxicology, these are the key properties that have been observed by other authors as well. Oxidative stress amongst others, inflammation, fibrosis, and fibrosis-like lesions in animal study. That's something that we have seen. Genotoxicity is quite, the report on genotoxicity is quite abundant and quite comprehensive in that context. Tumorigenesis, there are only few reports on that. However, we have to be cautious given that all the others are true. And that's the same for immunotoxicity. There are limited evidences of immunosuppression, but nevertheless, those studies are very well conducted to come to this conclusion. And as I mentioned, if we go back to the slide that we started with, the green highlights indicate at least one or more study, and in often cases highlighted by bold, multiple studies have shown similar changes for carbon nanotubes. So in terms of these evidences, based on the last IARC monograph meeting, where we stand is multi-wall carbon nanotube 7 has been classified as possible carcinogen to humans, which is group 2B, and multi-wall carbon nanotubes and others are not classifiable, and they are in group 3, but they are in urgent need of mechanistic evidence. That's where we are contributing to. And before I end this presentation, I have to emphasize this is the case with all the nanoparticles. There are multiple challenges that we face at a regular level. That includes assay interference, that includes interference with comet assay, micronucleus. The more regulatory accepted assays, there are consistent interference there. But we've, to a certain extent, overcome those challenges by incorporating different steps into the study design. And now we can have more or less reliable results. But of course, these results are often obtained from multiple tests and not one single test. And with this, I would like to thank the people who were involved in the carbon nanotube study, most importantly, Dennis O'Neill, who was my equal in this study. Always, we've worked together for the last few years. Lisa, of course, Dries and Tim, who were involved in the study with the telomere length and mitochondrial copy number, and Lode and Peter. I can't say thank you enough to all of you guys. Thank you so much. And with this, I would like to end my presentation. Thank you.
Thank you, Manosji, for the very nice and interesting work that you did. It was a very nice overview. Usually we don't do questions with award lectures, so I'm sure that there might be people in the audience with burning questions. But Manosji is still here for the rest of the session, so you can ask him questions in the chat directly to him. Also, the presentation will be made available online. Also, everything is filmed, so in the next weeks we will also put this on the YouTube for people to watch again. So if there is burning questions in the audience, do please chat with Manosji about this. So congratulations once more, Manosji.
Thank you so much.
Thanks for the nice overview of your work.